Welcome back. So before we went to the ad break, we were speaking about how cells give rise to organisms. So I want us to look at the picture alongside here. So as you can see, we have a muscle cell here. This is our muscle cell, and this muscle cell now grouped together with other muscle cells, as you can see on this picture, they form what? Muscle tissue, just like we explained earlier. And now these tissues grouped together, they form the heart, which is an organ. And now this organ grouped with other organs in the body of an organism, then they give us a full organism, in this case, our giraffe. So that is exactly what this slide was trying to explain to us. So this is another diagram that explains it further. So we have muscle cells grouped together, giving us tissue from the stomach. So now we have our tissue. This tissue with other tissues grouped together give us the organ, which is the stomach. And now this stomach grouped together with other organs like the brain, like your heart, your lungs, and everything else, then they will form part of the system or the body systems in your body as an organism, and that is how we end up with a whole human being. So here they say the stomach forms part of the digestive system and other systems in the body, forming an organism. So that is what we were explaining. So we are going to move on to the third concept, which is the microscope and the history and discovery of it. So when we are saying micro, what we mean is small, and scope means to observe or to look at. So microscopes are instruments that are used to look at or study objects that are too small to be seen with our naked eye. That is why we need a microscope to look at them. So Robert Hooke, who was the first cytologist to identify cells under the microscope in the year 1665, he decided to call microscopic shapes that he saw under the slice of cork, which is the cells, because the shapes reminded him of the cells or the rooms that the monks in the nearby monastery lived in. So that is how the term cells was coined by Robert Hooke, who decided that what he was seeing under the microscope actually reminds him of the rooms that the monks used to live in, and that is why he called them cells. So since the days of Hooke's observation, the development of microscope has come a long way. We have had many different types of microscope. Today we have the incredibly powerful microscope called an electron microscope, which uses electrons instead of light to observe very fine details. So we have two types of microscope. We have a light microscope and an electron microscope. So the electron microscope differs from the light microscope in that it produces image of the specimen by using the beam of electrons, whereas the light microscope uses the beam of light. So let us look at how they look. So this is the light microscope. This is how it looks. And then we have our electron microscope. So the electron microscope is the modern one and the more recent one that we use. So this is the one that uses electrons instead of light. Now let us have a look at the difference between the basic light microscope and the electronic one. The basic microscope allows you to see details in specimens that cannot be seen with the naked eye. The image you see needs to be well lit, which means it needs to have enough light provided. Also, it needs to be well focused and contrasted with its surrounding for you to clearly see the details. Obviously, if you need a microscope to see it, that means whatever it is that you are observing cannot be seen with the naked eye. So that is the power of the microscope. It allows us to see things that we cannot look at with our own eyes. The next image explains the different parts of a light microscope and what the function of each part is. So as you can see, this is our microscope. Now we are going to look at each part and its function. So the first one that we are going to look at is the ocular lens, which is also known as the eyepiece. So I think from the name you can already tell that this is the part that you put your eye on. So now the cylinder containing 2 or more lenses. So it has 2 or more lenses. These lenses are held at the correct working distance, and the lenses or eyepiece helps to bring the object into focus. That is the function of the eyepiece or the ocular lens. It is to bring the object to focus, and also this is the lens that you use in order to look through the microscope. The second part that we are going to look at is the coarse adjustment screw. So from the word adjustment, you can already tell that it has to do with limiting. So the coarse adjustment screw is used for the initial focus of the object. So if you want to put the object in focus initially, that's the first part that you need to use. And the second one is the fine adjustment screw. So this one is the coarse adjustment screw that is used for the final clear focus of the object. So if you want to see the final clear focus of the object, now before you use it, obviously you need to use the coarse adjustment screw. And when you want to get the final detail of how the object looks like, then we use the fine adjustment screw. And then we have our frame here. So the frame is a rigid structure that is used for stability. The frame is supported by the U-shaped foot leading to the base of the microscope. When they're speaking about the U-shaped foot, this is what they mean. This is the U-shaped foot or the base of the microscope. And then I want us to move to the opposite side. So we are going to look at the revolving nose piece. The revolving nose piece holds the objectives in place so that they can rotate and can be changed easily. So the objectives are these 3 parts over here. So the objectives magnify the objects. This is what the objectives do. They make whatever microorganism to look bigger so that you can see it with your eyes. There are normally 3 of them present, and all 3 of them are magnifying at different levels, as you can see over here. And then we have our stage. So the stage of the microscope is where you place your microorganism or the slide. So here the stage contains a clip. As you can see, these are your clips over here. So it contains clips to prevent the slide from moving around. So these clips are there to hold the slide on the same place so that whatever specimen you're observing, it doesn't move around and you can get a clear picture of what they look like. And then the last one is our diaphragm and condenser. So these 2 control the amount of light which passes through the slide. So this is our diaphragm over here. And where does the light come from? The light comes from this last part, which is our light source or the mirror. So if you can see, this is our mirror over here. So the function of the diaphragm is to minimize the amount of light that enters so that you can be able to have a clear vision of what is happening on the slide or the stage of your microscope. So those are the parts of your microscope and their function. We are then going to look at microscopic organisms and macroscopic organism. You need to be very careful when pronouncing those because they almost sound similar. So when we are speaking about microscopic, these are organisms like bacteria, which have a single cell. If you remember, we said if it has a single cell, that means it is unicellular. So the individual cells perform all the life processes for that microscopic organism. So basically, bacteria have only one cell, and that cell is responsible to keep the whole bacteria alive. So here in the picture, we have your examples of bacteria. They obviously have different shapes and different sizes, depending on what they do. or what they are specialized to do. So what about the cells in the macroscopic organism? So when we speaking about macroscopic organisms like human beings and animal, they consist of large numbers of cells, obviously because they are big. Remember, bacteria cannot be seen with our naked eyes. But when we are speaking about macroorganisms, these are big. These are human beings, these are animals. Obviously, you can see them with your naked eyes. Like the picture over here, we have a male and a female over here. So we call them multicellular organisms because they are made up of multiple cells. So that is why we call them multicellular organisms. We have already learned about specialized cells in macroscopic organisms. We know that not all cells perform all processes or functions. They are specialized to perform a specific function. For example, a skin is made up of epithelial cells, and the nerves, which is the yellow lines here, are made up of nerve cells. So what we mean in this point is that the epithelial cells in the skin cannot exchange roles with the nerve cells. Now we are going to move on to the last part of our lesson, which is our stem cells. So stem cells are cells that have the ability to divide and develop into many different types of cells. Their responsibility is to replace damaged cells or worn out cells, as they have the ability to develop into any kind of cell. So stem cells are divided into two forms. We have embryonic stem cells and adult stem cells. You will later learn about these as you do life sciences. Stem cells are quite amazing as they can divide and multiply while still keeping their ability to develop into different types of cell. So what do we mean by that? When we look at this picture, we have stem cells over here. So these stem cells have the ability to multiply and develop and become nerve cells. They can also become intestinal cells. They can also become blood cells, muscle cells, liver cells. So stem cells are normally used in research and in medicine to help people, especially people with cancer. They use stem cells to regenerate their dead cells or worn out cells or damaged cells. So that is what stem cells are used for. So we have reached the end of our lesson for today. Thank you for joining us, and I'll see you next time.